This is Mr. Worski again. This is the second screencast in excretion. We ended with this one last time. It was really quick, but we went over how the Bowman's capsule went to the proximal convoluted tubule, which was surrounded by the peritubular capillary, and from the proximal convoluted tubule to the descending loop of Henle, which moves to the ascending loop of Henle, and those are in the medulla. It moves back to the distal convolute, the loop of Henle is surrounded by the vasa recta. The ascending loop of Henle will become the distal convoluted tubule, which is also surrounded by the proximal peritubular capillary in the cortex. The distal convoluted tubule will join a collecting tubule, which will combine with other collecting tubules in the medulla and form the pyramids. Eventually, all the collecting tubules will join together to form the ureter. That's the general path. Now, our job is to go into much greater detail about what happens and what moves and how it moves and where it moves. So we're going to start with the Bowman's capsule. So remember that glomerular filtration rate? The blood pressure forces the fluid from the capillaries into the Bowman's capsule. That is known as the glomerular hydrostatic pressure. The more glomerular hydrostatic pressure, the greater the glomerular filtration rate. So now the fluid that is forced out will travel to the podocytes, which is the transport epithelia. Now, small molecules can enter the Bowman's capsule. Large molecules don't. If you have large molecules like proteins in your urine, you have a problem with your kidneys. You really do. The podocytes are an example of cells that are joined together by tight junctions. So the materials have to move through the cells, not between the cells. So remember, the higher the glomerular hydrostatic pressure, the greater the glomerular filtration rate. So now we have all these things, and here is the Bowman's capsule. Here are the capillaries here. The podocytes is between the capillaries and the Bowman's capsule, and the stuff is being forced out of here through the podocytes into the Bowman's capsule. And this is the proximal convoluted tubule. In the proximal convoluted tubule, the transport epithelium, many substances will be exchanged with the peritubular capillary. The hydrogen ions, and you might need to make note, and you're going to have to draw pictures of this, but hydrogen ions will move into the proximal convoluted tubule from the peritubular capillary. And those P's, C's, and T's are all crazy, aren't they? Now, this helps maintain the pH of the blood. Cells from the proximal convoluted tubule will also produce ammonia to keep pH of urine constant. Now, there's a range. I think it's anywhere from 6 to 8 that pH urine is found. Right? So drugs and poisons are also secreted into the proximal convoluted tubule from the peritubular capillary. Now, 99% of nutrients, glucose and amino acids, are moved from the proximal convoluted tubule to the peritubular capillary by facilitated transport. So the nutrients ended up in the nephron, then moved back into the blood by facilitated diffusion or facilitated transport. And here's the peritubular capillary. Here is your proximal convoluted tubule here, and this shows what moves back and forth. A lot of stuff moves back into the peritubular capillary because that's what we talked about, right? We talked about how 178.5 liters of filtrate is reabsorbed back into the body through this nephron. We have potassium, sodium, magnesium, phosphate, and sulfate. Water, bicarbonate, and other salts are moved back into the peritubular capillary from the proximal convoluted tubule. Now, 70% of the water, 75% of salt, and 90% of the bicarbonate is reabsorbed there. So about 60 to 70% of the filtrate that enters the nephron through the Bowman's capsule is reabsorbed back into the blood at the proximal convoluted tubule. So now, the descending loop of Henle, there's only one thing that the descending loop of Henle is permeable to, and that is water. So the water will enter the tissue surrounding the descending loop of Henle, which is hyperosmotic. So the water enters the tissue, and then that water will go back into the vasa recta, and that's your blood. The ascending loop of Henle, there's two sections. There's a thin and then there's a thick section. Now, the transport epithelium is only permeable to salt, NaCl. In the thin section, salt to diffuse out into the medulla, which help maintains the hyperosmotic environment because the water is flowing out in the descending loop of Henle. The salt is moved back out in the thick section by active transport. Eventually, the salt is going to enter the vasa recta. And here's the loop of Henle. This is the descending where water is coming out, and then the ascending when salt is coming out. And that helps maintain the osmotic environment, and the salt and the water are absorbed by the vasa recta, which is your blood. The distal convoluted tubule is similar to the proximal convoluted tubule, where a lot of things happen or move in this particular spot. The transport epithelium is going to allow for a variety of substances. Potassium and hydrogen ions move from the distal convoluted tubule to the peritubular capillary. We have salt, and that should be bicarbonate H capital CO3, moves into the peritubular capillary by active transport. Water moves back into the peritubular capillary by osmosis. So if we decrease the salt reabsorption into the peritubular capillary, we increase the amount of water that stays in your nephron. And if the water stays in your nephron, you're going to pee more. So if you like coffee, that's what caffeine does. It inhibits the movement of salt back into your blood. And since salt doesn't move back into your blood, the water stays in your nephron because the salt is in there, and it just increases the amount that you urinate. I got that already. You wrote that. Oh my gosh. Okay. And calcium moves back into and out of the peritubular capillary, and that movement of the calcium is affected by hormones, which we'll talk about in the next system. So keep that in mind, right? And here again is your distal convoluted tubule. Opposite distal convoluted tubule here. This right here is your peritubular capillary, and it shows the movement of some things, and that's kind of crazy. You don't have to worry about that much. The collecting tubule, water moves by osmosis into tissue, and then it moves back into the vasa recta. So urine can also move into the medulla to help increase the solute concentration of the tissue. Now, the urine will also go back into the vasa recta at some time and eventually be filtered out again. And here's the collecting tubule right here. Salt goes out, urea goes out, water goes out. Remember, all we're doing is I'm trying to increase the absorption of water. And that happens when we remove salt because of osmosis. It's all basic stuff. So how do we regulate the kidney? Well, the glomerular filtration rate is affected by blood pressure. And if we need to have adequate blood pressure or GFR for the kidneys to function, a drop in blood pressure will decrease the GFR and your kidneys will not function that well. So hormones will affect the kidney because we need to maintain a balance. ADH, which is called antidiuretic hormone, is produced by the hypothalamus in your brain and stored in the posterior pituitary, which is also in your brain. The hypothalamus is a major sensor of your body. It's an incredible gland, and it monitors the homeostasis of your body. It monitors blood solute concentration. When the solute gets too high due to excessive water loss, ADH is released and it targets the distal convoluted tubule and the collecting tubule, and it increases water absorption back into your body. Now, alcohol will inhibit ADH, and so then you're not going to increase your water reabsorption. In fact, you increase your water loss, and alcohol can cause dehydration because it inhibits ADH. So ADH increases water reabsorption, increases the blood pressure and will increase your glomerular filtration rate. The juxtaglomerular apparatus, thank goodness it has JGA as its initials, is located where the arteriole meets the glomerulus. So when sodium is low, the blood pressure is low. So angiotensin releases an enzyme called renin. Renin will activate angiotensin, which is a plasma protein, and it changes it to angiotensin 2. Now, the angiotensin 2 will stimulate the adrenal gland to release aldosterone. Aldosterone targets the proximal convoluted tubule to increase salt reabsorption into the peritubular capillary. And as you know, if you increase salt reabsorption into the peritubular capillary, water is going to follow it and therefore increase water reabsorption as well as your blood pressure. You increase blood volume, you increase blood pressure. So this is the second way that you know of, along with ADH, to increase water reabsorption into the blood. So in order, and here's the JGA. I don't know if you remember this, but right here is where JGA is, and right between here. This is your arteriole. This is your Bowman's capsule. That's the JGA. Third type is the atrial natriuretic factor, or ANF. Now, in order to maintain homeostasis, you have to have something that works opposite of the other two, right? So if something increases blood pressure, you need to have something that will decrease blood pressure, and that's what ANF does. It opposes the JGA. The cells are found in the right atrium of your heart, and when you have high blood pressure, when you have high blood pressure, your ANF will be produced, and that will inhibit JGA, which inhibits the release of renin, so that will bring down the water that you absorb, and that will decrease your blood pressure. Remember, homeostasis, you need that balance. Well, we are done with the second screencast. If you have any questions about the information that we went over in any of the screencasts, by all means, you can either watch the videos over again, you can read your textbook, or ask me a question tomorrow in class. Have a good evening.